I'll go ahead and introduce Matt. Matt Tancos is a research plant pathologist with the United States Department of Agriculture, Agricultural Research Service in Fort Detrick. He's with the Foreign Disease Weed Science Research Unit. He has Ph.D. in plant pathology and plant microbiology from Cornell University. He researched bacterial plant pathogen, Rathayibacter toxicus, while a USDA-ARS postdoctoral researcher. Matthew's research program is currently investigating the use of plant pathogens as biocontrol agents to manage invasive weeds in the United States, characterizing pathogen emergence and evolution in our agroecosystems, and investigating novel molecular-based invasive weed management strategies. So I think at this point you could take it away, Matt.
Well, thank you for that, Jill, and thank you for extending to me this invitation to speak to you all today. So today I want to tell you about some of the exciting research that we're doing with the United States Department of Agriculture, and specifically at the Agricultural Research Service. So I am actually based at the Foreign Disease Weed Science Research Unit, which is located in Frederick, Maryland at Fort Detrick. So again, my name is Matt Tancos. I'm a research plant pathologist. So today's talk is going to give you an overview of what we're doing at our facility, the research we're doing, some of the weeds we're investigating, and a big picture overview of the regulatory hurdles and processes that go into finding a potential microbial pathogen for biological control into getting federal regulatory approval to release it in the United States. So this should be an all-encompassing overview. Please feel free to ask any questions at the end of the presentation. So I think everyone here knows the importance of our native plant communities, our natural ecosystems, and the impact that non-native invasive weeds have on this. So invasive weeds, non-native invasive weeds, directly impact our agricultural systems, our natural and urban systems. And just in our agricultural systems alone, invasive weeds cost the United States $25 billion in losses and damages, plus an additional about $10 billion in control costs every year. And invasive weeds, they compete for limited resources. They injure livestock, both physically and also through chemistry and toxins. Invasive weeds also act as reservoirs for plant pathogens and pests that can jump into our agronomic crops and systems and livestock. They also displace native and beneficial plant communities, and they can also choke irrigation systems. And this is especially important out west, where water can be a very limited resource. So I actually included two images of grazing livestock because rangelands and forests and some of these large terrain areas are some of the most difficult terrain to manage due to their size, topography, and diversity of plant species. So in fact, rangelands alone consist of about 770 million acres of land. So that's about 30% of total US land cover in the US. And invasive non-native weeds in these rangelands cost landowners and land managers about $6 billion every year in damages and management costs. However, we know invasive weeds go well beyond just impacting our agricultural systems. They also directly impact our natural and urban systems. So we know the importance of our natural systems. They're vital because they feed both directly into our agricultural and urban systems and are truly priceless in value. Whether they're pollinators, natural communities. We also have diminished water quality and quantity from invasive weeds. That also impacts our recreational use and associated businesses, whether aquaculture, tourism, recreation. And invasive weeds also play a big role in exacerbating wildfires. And we continually see this in the news, especially out west. Seems like it gets worse every year with the wildfires and how frequent they are and how severe they're becoming. And invasive weeds really take advantage of that because it's abundant fire source, fuel source, and also they're very quick to grow back and reestablish and take over these disturbed areas and then further cascade that issue. And lastly, as I mentioned, invasive weeds can harbor plant pathogens that can be detrimental to our agricultural systems. They can also impact human and animal health by harboring pests that can carry and transmit pathogens, whether for humans or our livestock. So, for example, the CDC currently recognizes 18 tick-borne pathogens, and Lyme disease alone is costing the U.S. about $1.3 billion every year. So invasive weeds truly impact every facet of American life, and it really impacts our natural resources and issues that are vital to our national security. And you don't really have to be a grower to see the impact of invasive weeds. However, with biological control, and that's either using microbes such as plant pathogens, which is what I am hired to do, or insects, this can be a cost-effective approach to combat invasive weeds over large, inaccessible, or ecologically sensitive terrain. So I wanted to give a quick overview of our research unit. I'm assuming there's a lot of individuals here who have not heard about our research unit or what we're doing. So I just wanted to put everything into context for everyone. So again, I'm at the Foreign Disease Weed Science Research Unit. We're located at Fort Detrick in Frederick, Maryland. And Fort Detrick is an active military garrison. I'll get into that on the next slide about the history of why the USDA-ARS is at a military base. But ultimately, our research mission is unique in that we are mitigating the spread and establishment of invasive pests that threaten the United States agriculture, our national security, and our natural resources. So our unit is kind of split up into two research areas. We have individuals that are investigating invasive plant pathogens that are detrimental to our agronomic systems and crops. And then we have the group that I'm leading involved with invasive plants and better understanding the diseases that are associated with them. So both research areas are investigating plant pathogens, and we both utilize our unique biosafety level 3 plant pathogen containment facilities, but with different end goals. So my colleagues are trying to better understand plant pathogens, either emerging pathogens or exotic foreign pathogens, such as select agents that are not in the United States yet, but pose a severe risk to our agriculture if they are ever introduced. So they're looking at understanding the pathogens, developing diagnostic tools, and tools that can help mitigate the spread if they are ever introduced. On the flip side of that, I'm kind of doing the opposite. So my goal is actually to identify these pathogens and increase endemics and outbreaks. However, I am trying to control these non-native invasive weeds. So I'm trying to cause these outbreaks and disease development in these non-native invasive weeds. But my colleagues, they're investigating phytopathogenic bacteria, viruses, fungi, and oomycetes. And then the group that I'm leading, we're investigating diverse weed management strategies, both classical biological control, but also new techniques using genetic biocontrol tools and molecular approaches, doing disease development, optimization, and then also looking at pathogen evolution in the role of invasive weeds in harboring plant pathogens and viruses. So one of the questions I always get is, why is the Department of Agriculture at a military base? The reason we are at Fort Detrick has a lot to do with the history of this research unit and the facilities that we maintain. So originally in 1942, in the midst of World War II, the US, like other nations, were beginning to develop offensive biological weapon program. So ours was actually initiated at Camp Detrick in Frederick, Maryland. And then from 54 to 1971, it was actually part of the US Army's Plant Sciences Directorate. And then in 1969, President Nixon ended the US biological weapons program. So by 71, all of these facilities and high containment facilities were transferred to the United States Department of Agriculture. And that became the Plant Disease Research Laboratory. And then by 1979, it became what it is today, part of the Agricultural Research Service, or ARS, and the Foreign Disease Weed Science Research Unit. And so that was a little background about why we're here. And we're here because we have these high containment research facilities that were originally designed and developed for the idea of, like I said, biological weapons. But after all of that was abolished, now it's for researching and keeping organisms out of the United States. So our facility is unique that we have a 10,000 square foot biosafety level 3 plant pathogen containment facility, which includes a greenhouse and laboratories. This is actually the largest BSL-3 containment facility in the United States that's approved by the Animal Plant and Health Inspection Service, APHIS, for research on exotic plant pathogens. So currently, USDA-ARS maintains 4 of these BSL-3 research facilities. However, 3 of those are entirely focused on animal diseases. So we are the only ones that are focused entirely on plant diseases. So this facility has research laboratories, we have greenhouses, growth chambers, dew chambers for these inoculation experiments, and also a variety of instruments and tools for both molecular and applied research. And this entire facility is under negative air pressure to keep stuff in. All the air is filtered through HEPA air filtration multiple times every hour. We have our own wastewater treatment facility on site to make sure nothing is escaping in any way. All products and plants are completely autoclaved and sterilized before exiting, and only authorized personnel are allowed to enter the facility. And then we have to actually put on clean scrubs that we wear during our research. And then when we exit, we all have to shower so we don't bring anything out with us. And this facility is really unique. It provides us unique opportunities to investigate some of these invasive and emerging pests in a greenhouse setting while still under high containment. So it gives us a lot of opportunities to really explore some of these relatively unknown pathosystems and hosts that they may be targeting, both for invasive weeds and in the agricultural section as well. So specifically for my group, the Weed Biological Control group, like I said, we're investigating invasive weed management strategies, optimizing disease development, and better understanding pathogen emergence and evolution. So to give everyone a better idea of how our unit or how our lab, our group is broken up, is we have really 3 research missions. The first is the conventional approach, and this is more of the classical biological control. So this is involving pathogen exploration, both endemic pathogens native to the United States that infect these invasive weeds, but also foreign exploration because in our group historically predominantly focused on the foreign aspect because the idea is that you go to the native region of where these weeds evolved and developed to try to find pathogens that they co-evolved with. And when they were introduced, a lot of the time they don't bring anything with them. And that is one of the reasons they can be very successful and outcompete. So the idea is that we can then bring one of these pathogens that evolved with them and kind of exert that negative pressure on them to suppress their population growth and ability to rapidly spread. So the idea of biological control is not to eradicate them because that's going to be very difficult, if not impossible, but ultimately to control the population and make them less invasive. The best tool to prevent invasives is to keep them out because once they are in here and they start spreading, it's very difficult to completely eradicate them. So anyways, foreign pathogens and endemics, and then we have to evaluate those. We have to make sure they're efficacious, that they're going to cause disease, but also that they're very specific to the invasive weed that we're trying to kill or target in this case, and then trying to optimize our inoculum, our pathogen, to increase disease development so it can really hamper the population once it's released. So that's the conventional approach. We are also doing a molecular approach. So this is more of the basic research, looking at one of the things I'm really fascinated about is the role of invasive weeds as reservoirs, especially all the microbes that they bring over with them. So, and not only do you have this foreign plant being introduced and spreading, But what about all those microorganisms and viruses that are present within that plant that are also spreading? Those are all novel biological systems, potentially. So what role are they playing? And any organisms that do develop pathogenicity against these weeds, what role are they playing in interacting with our more better understood agronomic pathogens that can infect our crops? Is there any gene transfer occurring in pathogen evolution to make more aggressive strains? So that's one area of concern that we're trying to evaluate. We're also looking at the microbiome and virome of these invasive weeds. And the last approach is our technology-driven approach. So wanting to really utilize molecular-based advances to augment our microbial-based biological control agents. So this really falls into genetic biocontrol tools and new technologies that we can evaluate, determine their efficacy and safety in our high containment facilities. However, for today's talk, I am just going to talk about our conventional approach, which is the pathogen exploration or the classical biocontrol. I won't go into the other stuff, that can be their own talks, but just to give you a better idea of all the research that we are doing in our group. So to give you a little better understanding of the process that goes into identifying a promising potential biological control agent that can be released into the field. It's actually a very complex, long time process to get it all the way to be federally approved. And I will go into the federal regulatory overview at the end, just to, again, give you a better idea of what all goes into this process. So here's a quick breakdown of what happens and how we go about this with the classical approach. So first we have our diseased plant tissue from our invasive plant. These can be endemic samples that we collect in the US or from foreign collections. First of all, you have to isolate and identify your pathogen. Depending on the quality of the tissue, sometimes these plants can be readily degraded. And then you have other organisms called saprophytes that can just start degrading the material, and they're kind of secondary, but they're not the ones that cause the disease. So you have to isolate all of these microbes, and then you have to evaluate and determine which one's the actual pathogen. So first you have to do the isolation identification, and then we go down to step 2, and this is to confirm you have the pathogen once it's identified. Well, before you go through this whole process that can take years, you want to make sure you're putting all of your focus on a promising candidate that's going to be efficacious. It's going to cause a lot of disease and suppress population growth or seed production. If the pathogen is just an okay pathogen, well, maybe you drop it and you have to go back to step 1. If it is a nice pathogen that has potential, then you go to step 3. And this is actually the most time demanding portion of this. And this is to demonstrate that we have a specific pathogen, host specific, that is only going to infect the non-native invasive weed that we want to control. Because, and this is very important, because we don't want to, especially if it's a foreign microorganism, we don't want to bring something in and release it and then have it infect something else that it wasn't meant to infect. So a lot of work and a lot of regulatory oversight goes into this to ensure that we have a organism that's going to be very specific to just this non-native invasive weed. So if everything goes with it's efficacious, causes disease, it's very host specific to our invasive weed, then we can go to step 4, where we put all of this together in a petition that then goes through a regulatory review process. And again, if they approve it, then we can go through the process of getting permits and applying it. If they reject it, you go back to square 1. So that's just a quick overview of that. So if we get into, so what are some of the invasive plants that we're targeting? So we actually look at a diversity of invasive plants. And one of the difficulties of this is that right now our group is only myself as lead scientist. I have a colleague, Mike Fulcher, and then we each have a technician. So it's 4 of us. And you know how many invasive weeds there are in the United States, let alone in just the mid-Atlantic region. So there's a lot of diverse stakeholders that all have their top 10 invasive plants, whether it's all the way out west, down south, in the east, or Midwest. So one of the challenges we have is to balance our research to benefit our diverse stakeholders from across the United States. But our current research areas are really are focused more on the Western U.S. rangeland invasive weeds, and then also the Mid-Atlantic and Eastern invasive weeds. And here are just some images. Obviously, we know the diversity of environmental conditions. So this is out west in Idaho, a lot of rangeland, mountains, diverse ecosystems. And then out east, this is actually fig buttercup in the forest understory. Again, very diverse environments and systems we would be evaluating. So I briefly just want to, again, give you an overview of all the stuff we're doing. So the first, I just want to discuss briefly, since this is involving Maryland native plants and plants of Maryland in the Mid-Atlantic, but a lot of the research and time we put in goes to some of these rangeland weeds. And the first one is actually common Croppina. So this is actually an Asteraceae. It's a federally listed noxious weed, and it was listed in 1968. It's an overwintering annual weed with origins in the Mediterranean region. It's a close relative of knapweeds, and it competes with grasses and forbs in grazing and natural areas. And it really reduces rangeland productivity, which is a big concern over there, or out west. But currently it's only identified in Idaho, California, Washington, and Oregon. In here, I just have some images of these infestations. I still have yet to see this plant in person in the field. I'll discuss this in a little bit, but it's a truly hardy plant. So in this first image here, I hope you can see my mouse. Here's the plant right here, flowering, and it's incredibly hardy. It's growing essentially out of this cliff rock side. And this is one of the big areas that makes it difficult to control. Even if you, just the terrain that it occupies, conventional herbicides or cultural tools aren't really going to work at this scale to manage these invasive plants. So here's this growing in a cliffside. And then here's actually, this is, I believe, in Hell's Canyon in Oregon. And all of this is the noxious weed, Croppina. You can see some of it up close. And then over here is actually in Idaho. You can see another large infestation of the Croppina. It's a relatively inconspicuous weed, but it really takes off. And that's one of the reasons it's spread so far, is that it is inconspicuous. It doesn't really stick out until it's too late and you have a whole area colonize. So here's another image of this valley of just complete coverage with Croppina, really spreading. Here's another image. And this is an Idaho infestation near, this is Salmon River. And again, complete infestation over here, outcompeting all the native plants. However, we were fortunate that we actually finally just got federal approval in late December of 2020 for a fungal pathogen, Ramularia, that was originally identified in southern France. So this is a foreign one that had to go through all of these processes in our containment facility and then be approved at the federal level for release in the United States. This is a stem and leaf infecting fungal pathogen. So here's some of the symptoms you see on a plant. You can see the lesions developing. Here's some of the spores of this fungus that allow it to spread. It's very host specific, and there's multiple genotypes and varieties of this invasive weed, and this pathogen can actually infect all of those that are known to exist in North America. So we actually have stuff ready to go to do some of these initial field trials to see how well it's going to perform in an actual field setting as opposed to a greenhouse. So here's another image of some colleagues from the University of Idaho and also ARS. And this is actually in the Nez Perce Reservation in central Idaho. And you can see it's a beautiful area. And these are setting up some of the field plots to do these inoculations. So since this weed is a winter annual, it germinates, quick overview, it germinates in mid-fall, late fall, and then it overwinters in a rosette close to the soil. And then you have all of winter, it kind of stays dormant. And then following winter into spring, that's when the plant begins to grow more, and then it bolts and pushes up a flower. And then you have flower and seed production in May and June. And then by the end of June, the plant's senescing and it's dead. And then the life cycle completes again. So we're actually going to try to inoculate both in the springtime when it's going to take off and flower, and then also in the fall time, because the fall is a very conducive conditions for disease to develop. It's very warm still. The soil is warm. You still have a lot of dew and rainfall. So it's just a really pristine time to get disease to develop under these conditions out west. So we had sites selected within the Nez Perce Reservation and other parts of Idaho for our preliminary releases. However, we had everything ready to go at Halloween time this Halloween, this October. And then the West got nailed with a big cold front, early, unexpected winter system that came in, dropped a lot of snow and a lot of hard freezes and frosts. And that essentially is not going to allow this pathogen to infect. It's not going to work when it's in a refrigerator. But and these are my colleagues just to show the snow they were getting. So we had to pull it off. So we're going to postpone this until the spring and then try again next fall as well. But we were able to at least get some of the weather stations put out so we can have a better idea of what the local conditions are for the coming releases. And that's just some of the difficulties with field research is, especially with all the variations in weather, you can never predict it. And you get these extreme events that are unexpected. But the second and the last rangeland weed that I'm going to discuss is Russian thistle, or AKA tumbleweed. So before I came into this, again, I'm a plant pathologist. I was totally surprised that tumbleweed was not a native plant. I always see it in Western movies. It's kind of a hallmark of the Western US, but this is a nasty invasive from Eurasia. It was introduced in the 1870s, believed from contaminated flaxseed. And this has a rapid dissemination. A lot of seed can be released. And especially when it breaks off at the soil line, this plant can tumble and you spread seed even quicker and you can spread it at great distances. It also has a high tolerance to dry climates. And any little rain or water will allow this plant to really take off. And here's just some images of this. These can get incredibly large. And this also reduces rangeland productivity, but also is a huge fire hazard because there's even videos on YouTube where you can see these giant tumbleweeds on fire rolling across the plain. So nasty plant all in all. And right now we are in the process of finalizing some work on another fungal pathogen. This is a rust species, a rust fungal pathogen. And this was actually identified in Russia. Again, a 4-in-1, more from the native region, trying to identify some of these specific pathogens. And this causes leaf and stem infections. So this image here is a young Russian thistle. And you can see these lesions developing on the stems and the leaves. And here's an up-close image of that, where you can see these pustules erupting. And all of these little orange powder-looking stuff are the spores that can then start additional infections. And it's called a rust because of the color. It has that rusty color from these spores. So that's where it gets its name. And then as the disease progresses, here's a later image of that where you have plant death, and then it's really just severe infestation. And during this stage, it actually switches over to a different spore. These are the Telia that are more overwintering spores, and they lose that orange rusty color. But anyways, we have some work to do for the Fish and Wildlife Service. They wanted us to do some more studies on a couple threatened and endangered relatives in North America. So we need to identify those and test them before this agent can be approved. And that's one of the difficulties with this is if you have to evaluate with using against threatened and endangered plant species to ensure that they won't infect them, It's not like we can just call up our seed grower and get some of these threatened and endangered plant species to evaluate in our greenhouse. A lot of the time you have to have the contacts, you have to go out there to find the plants, get enough seeds so you can even do these experiments. So that's one of the challenges that I've identified in this, is that getting access to all of these plants is a challenge, and then getting them to grow in a greenhouse is its own issue, because you'd be surprised how well these invasives perform outside, but in a greenhouse or a growth chamber, they do not like it. So it takes a lot of time to troubleshoot that to make them happy, so then you can do experiments with it. So that's all I'll say about the foreign foreign pathogens for our Western rangeland weeds. And again, it was just to give you an overview of the diverse projects that we're doing and what we're looking at and how it progresses. For the remainder of the talk, I'm going to discuss some of the Mid-Atlantic and our Eastern invasive weeds and some of the weed targets we're investigating. So here's a list of some of our targets that we're currently looking at. Either we have pathogens for them. Now, whether they're going to be a promising agent it's still too early in the process, but we're finding pathogens, or these are some agent or weeds that we would like to identify some pathogens for. So we have stiltgrass, Perilla mint, garlic mustard, fig buttercup, wavy leaf basket grass, poison hemlock, Japanese hop, swallower, knotweed, and honeysuckle. So I'm not going to have time to go into all of these, but I wanted to pull out a few to give you a little more idea of what we're doing. So we're going to focus on the stiltgrass, Perilla mint, garlic mustard, fig buttercup, and wavy leaf basket grass, and give you a little overview of some of the projects we're doing and the different stages that they are in at the moment. So the first one is stiltgrass. I don't think this group needs a lot of background on stiltgrass. It's widely prevalent throughout our region. It's native to Eurasia. And while there are endemic pathogens that we've identified and other groups have, none of them, to my knowledge, have the potential of being a biological control agent that's really going to suppress populations on a large scale. So therefore, the U.S. Forest Service actually initiated a program with collaborators and colleagues in China to do some foreign exploration to try to identify some pathogens of stiltgrass over in China. And I believe they were also looking at insects as well. I don't believe any of the insects panned out. They found several different fungi, but the most promising ones that were specific and showed damage were actually some novel rust species. So some of those fungal rusts. So here's some images of that. And this is a slide from our colleagues in China. And there was a lot of promise to these. They had a lot of preliminary data that showed impact in China and their populations. So the U.S. Forest Service reached out to us to see if we would be able to acquire this rust, evaluate it in our secure containment facilities, ensure that it is specific and that it's going to work against all of our ecotypes and genotypes that we have in the United States. So we are currently working with the Forest Service, the U.S. government, the Chinese government, and our collaborators in developing agreements so they can transfer this to us to investigate. I'm an optimist, fingers crossed, that we'll be able to achieve this and get this over here so we can start investigating it and actually see if this is going to be worthwhile to take it to the finish line and take it to the federal overview process. So that's what we currently have going on with the stiltgrass, trying to get something that's going to be from its native region. Now, the next invasive weed that I wanted to talk about is Perilla mint. So this is actually the work that my colleague is doing, Dr. Mike Fulcher. And yeah, so Perilla mint is actually found in 20 states, approximately. Here's a map overview of it in the US. It outcompetes desirable plants in pastures and natural environments, and it is toxic to livestock. However, in 2021, last year actually, my colleague Mike, he was actually going around scouting areas in Maryland, and this was actually along the C&O Canal. And here's an image of, I don't know if this was the exact area, but something like this, where you can just see large infestations of Perilla mint. And he identified some nice disease. It's like here are some of the disease images of the leaves. He was able to isolate, identify the pathogen, and here's a pure culture of this fungal pathogen. So this is actually a Calotropicum species that has only been previously identified in Japan. And this was just recently published. I think he just published it a couple days ago. It was released. And this is the first time it's been reported in North America. And this pathogen actually produces 100% plant mortality under optimal disease conditions. So this has a lot of potential and a promise, but it's still in that stage. And in this case, it's an endemic pathogen, so it's not a foreign one, which makes stuff less complicated. But here are some of his data looking at this. So it's a very effective pathogen. So if we look here, so all of these plants were inoculated at the same time. So these three were actually inoculated with our pathogen, the Calotropicum species, and you see severe stunting and reduction of shoot and root length. And here are the controls that were just sprayed with water. You have normal, happy plants right here, this invasive. And again, these were all inoculated at the same stage, where you see this severe reduction when you have this disease and pathogen infecting it. Here's a more pronounced demonstration of this. You have our control plant that was inoculated with water versus our inoculated one that was actually infected with the pathogen. And these severe infections cause stunting, premature defoliation, reduced biomass, and reduced flower and seed production. And we kind of see that here in his data. So again, the controls are just the plants sprayed with water versus with the pathogen actually in the water. So we see a significant reduction in plant biomass when it's infected. We also see a significant reduction in flower production in seed when it is infected with this pathogen. And then he's also performed some competition studies with some native plants, so native mint species, mountain mint and bee balm. And he sees the same result with both. So one of the ideas, if we are able to suppress this invasive, can the natives then be more favorable and kind of take over and do better in that environment if they're not competing for resources against these invasive plants? So right here, I believe this is the bee balm, this example right here. So if this native plant is inoculated with their pathogen or the water, we don't see any difference from the water control. So the pathogen doesn't infect it. And then when he did competition studies, so growing Perilla with these native mint species, without the pathogen, you see a significant reduction of the native species, its biomass, because it's being outcompeted. But once you add that pathogen and really suppress the invasive, or Perilla mint in this case, now you see a lot of biomass from our native species. It's able to outcompete and grow now because the invasive was suppressed with the disease. Here's some more, some of the research that goes into this. So currently he's looking at different spore concentrations of what you would have to apply out in the field. So you have our control and then different concentrations of spores. And if you get it high enough, you will kill the plant right out within a week. And then we also evaluate dew periods. So how much water is required to get that initial infection. And here's the different hours. Again, if it's in there long enough under that optimal disease condition, you can just kill the plant outright. And again, another image of a control inoculated, just water, versus inoculated with the pathogen, you can have complete plant death. So current work is still being used to optimize this, develop the inoculum, how best to get infection out in the field. He also did some preliminary host range studies. He tested 10 commonly grown herbs from the mint family and closely related native mint species, but none of those have shown any symptoms. It appears to be very specific to this invasive Perilla mint, but again, that's going to have to be expanded even further. But he's hoping to do some ecological field studies this summer with the National Park Service in Maryland to see what this will do under more natural conditions. The next invasive weed that I want to talk about is garlic mustard. Garlic mustard is a biennial cruciferous herb from Eurasia. And again, this one's very prevalent in the mid-Atlantic in the eastern US. And when I say biennial, that means it requires multiple years to complete its life cycle. So usually the first year it's just growing just as a rosette, and then it requires that second year to then bolt and flower to produce seed to complete its life cycle. It was introduced in the 19th century, rapidly displaces native flora and modifies soil microbial communities, and actually identified some novel fungal and bacterial pathogens from garlic mustard in Maryland that I'll talk about shortly. Currently, we still have ongoing host range studies for both the fungal and bacterial pathogens identified, but also looking at doing genome sequencing and better understanding at the molecular level where these novel pathogens came from. Are there any known examples of these occurring in its native range? Is this a new pathogen that has exploited this niche in North America because you have a plentiful host being these invasives? So given enough time, do you see new pathogens emerge and new diseases develop? So that's some of the questions we want to address as well. So the first one that we identified was a bacterium, a gram-negative bacterium, Xanthomonas campestris, a novel isolate that it was identified to infect garlic mustard. And currently, the host range appears very limited to garlic mustard. We're still in the process to evaluate a variety of ornamental crucifers and other cruciferous plants, but none of the agronomic crucifers such as cabbage, broccoli, Brussels sprouts show any disease from this bacterium, which is very promising. So here's an image of this. We have our infected leaf with the bacterium, and then we have our control, which doesn't show any symptoms. But one of the questions that's interesting is, so the species of bacterium can infect a variety of cruciferous plants, both weeds and agronomic brassicas and cruciferous like the cabbage and that. So this strain is actually, this isolate is appears to be specific to the weeds and garlic mustard. But another interesting avenue of this is that there are related strains of Xanthomonas campestris that infect our agronomic brassicas, such as cabbage here. So one of the things I'm doing right now, and what we did with colleagues at Cornell University, is do some genome sequencing of these bacterium to see how they relate to the Xanthomonas campestris strains isolated from garlic mustard and other weeds. And do we see any evidence of pathogen evolution and actually transfer of genes in the development of new pathovars or different strains and host ranges? But I won't go into too much of that. Here's just a recent paper that was published, again, looking at the diversity of all of these strains, both from weeds and agronomic systems. And that's with a lot of these systems, just because you have a species, you could have a strain that's very specific, but it's still part of a species that can infect a variety of things. So you still have to do all that work to demonstrate that this given strain or isolate that is specific. So in this case, the Xanthomonas campestris, these are all the genomes of phylogenetic tree. So all of the pathogens up here infect the agronomic crops of brassica. Down here, these are more ornamental crucifers and different ones. So our weed isolate that looks very specific is down here, far away from the crop infecting ones. So that's some of the work we're doing with the bacterial pathogen of garlic mustard. Another one that we're looking at is a fungal pathogen. And this was fairly recent, just this spring, 2022 in Montgomery County in Maryland. I actually went out to the field. An individual was wanting to show me some symptoms, and we stumbled across this. We weren't even looking for this, but there was this one section where the garlic mustard was completely defoliated. And this is early spring. You're starting to see the seeds developing here, but this is way before the plants would senesce. In other areas further away, you had normal, healthy garlic mustard. But just in this vicinity, you had a lot of death And it's hard to capture here on the camera, but all you see are the garlic mustard stalks and what few leaves remain, and then leaves of other plants. So we were able to identify a few of these infected diseased leaf tissues, take them to the lab, do some isolations, and we were able to identify this new pathogen that truly it's pretty interesting. It kind of just liquefies the leaf, very aggressive. We're currently doing characterization studies and sequencing to identify what it is. Currently, it looks like it is a new species, but we want to do more sequencing and genome sequencing to confirm that. And then we're going to go on to host range studies and see, is this specific to garlic mustard or does it have a broader host range? But sometimes like this, it's more of just luck that you're in an area, you see something, and then you might find something from that. So this is very new. We're still in the active process of characterizing this fungal one. But now we have a bacteria, a fungal, and both of these are native or endemic. So we're trying to characterize those and see if they're a promising agent. The next weed I want to talk about is fig buttercup, another target that we're looking at. So this is native to Eurasia, introduced as an ornamental. It's in the buttercup family. And this is an early spring plant. And it prefers forested floodplains, and it reproduces asexually via bulbs and tubers. And this actually allows it to spread so rapidly because if you have flooding events, high rain events, these tubers can just spread and you really just get a dense carpet of this invasive. And overall, it's a very short growth period. It's one of the first plants to pop up in the spring, even faster than some of our natives. And for that reason, it really just outcompetes everything. And APHIS actually ranks this as a high-risk invasive weed. So in this image here, this is in Frederick County, you can see just a complete carpet of the fig buttercup in the forest understory. It even outcompetes garlic mustard, poison hemlock, and other invasives, but just amazing of how effective this plant is. And here's some of the classical heart-shaped leaves with the flowers. And also, this plant is toxic if consumed. So here's another image of just how dense this plant is, which is really just impressive to see how dense this plant can get and how happy this plant is. I scouted numerous areas and everything is always pristine, never any diseases, no blemishes. This plant is just happy and thriving, even at these high densities, which is impressive. However, I was lucky and stumbled upon some nice disease on one of the sites that was developing on the margins and is very localized. And this is actually, again, recent research that we were doing this spring. So I'm currently studying this, and this appears to be two different bacterial species. But as I've said, we have not had luck finding many pathogens of this. So I'm really excited about this one. We'll see how effective it is and how specific it's going to be. But we're currently investigating these bacteria and also some fungi that we found. But those are actually in the greenhouse as we speak, being evaluated. And then the last one I want to discuss is wavy leaf basket grass. This is another APHIS ranked high risk invasive weed. And this is an interesting system because it's a recent weed invader in 1996, identified in Maryland, originally from Eurasia. It's a rapid colonizer, spreads through seed movement, and it has very sticky seed that can adhere to fur of animals or shoes or pants and can be transmitted and spread that way. But the work we're doing right now on this is characterizing soil microbial communities from invasive wavy leaf basket grass invasions. And this is really being researched by my colleague, Dr. Mike Fulcher. He's working with Towson University and the University of Richmond. And to date, we've identified what appears to be some diseased tissue, but we have not identified any pathogens of this invasive weed. And again, this is a very recent invasion, the late 90s. So maybe it takes time for native pathogens, endemic pathogens to evolve to infect this plant. But currently we have not identified anything. So again, with any of these plants, if you identify diseased tissue or large areas that seem to be dead or die back, those could be very interesting sites to evaluate and look for some of these organisms. But for instance, in this case, maybe the wavy leaf basket grass might be a better target for foreign exploration. So it all depends. And then lastly, hopefully I don't go too long. The last slide is really looking at the overview of this regulatory process. That's very important. And it's important for you to know just how all the different individual components that go into this research, but also the importance that the oversight the US government is doing to ensure that we are only introducing safe and effective biological control agents, especially if they're from a foreign region. Because once something is released, there's no real bringing it back. So there's a lot of oversight to ensure that these are specific to the invasive weed that we want to control. So the first aspect of this is the agent evaluation and selection. So first you have to do the exploration, whether foreign or endemic. You have to do the efficacy testing, host specificity testing, and then put all that data together in a petition that you're going to then submit to regulatory agencies. So all of that data collection and exploring, finding the agents, especially for these foreign things, can take 3 to 7 years. And then the regulatory process, it actually goes to APHIS, and then it gets reviewed by this technical advisory group for biological control that we call TAG. This is an independent review panel that is comprised of members from different federal agencies in other countries. So we have Mexico, Canada represented, Bureau of Land Management, EPA, Fish and Wildlife, and other USGS, I think, is also on there. Variety of individuals that independently review these petitions, and then they make a recommendation to APHIS. So they can recommend for release or they do not recommend release. APHIS takes those recommendations, it performs its own review process. It either accepts or goes against the TAG suggestion. So if APHIS does approve it, TAG approves it, then it goes to the Endangered Species Act review. Following that being successful, it goes to the National Environmental Policy Act in tribal consultation. Following that, and if all that is successful, you have a work plan, then you have a regulatory publication, and then finally you can have a permit completion at the federal level. So all of that can take 2 to 4 years or more, and we've seen it take longer than that. Once you get through that process, you also have additional regulatory oversight that can come from the state level, county, and even park systems. So the National Park Service actually has its own review process that can add to time. And then you need those approvals. And then another big hurdle is the EPA, especially with microbial-based biological control agents, because these are considered biopesticides. So the EPA will review those as pesticides, and they might require additional data that you might have already performed with APHIS, or they require more of that. So again, this is a lot of processes that have to go through before you can release it. And the big issue with the EPA overview and approval, this applies to both foreign organisms and endemic. So even though I find something in Maryland, and say I find it, I deem it's effective and promising, I'm not just able to release it as a biological control agent because EPA needs to review all this. You have to show all of this data to EPA to get that approval. So it complicates things, but it is doable, it is feasible. It's just good for everyone to know the time frame and all the things that go into these processes. So even though we identify something in 1 year, it's not going to be something a year or two later. It's going to take time for all of this to occur. And then finally, if we get approval like we did with the Ramularia of Croppina, then we coordinate with collaborators and we can begin release in long-term studies. And with that, I'd like to thank you all for your time. I hope I didn't bore anyone, but gave you a broad overview of what we're doing, how we do it, and the individuals that are bolded are in our group. Jamie Thomas is my technician, Dr. Mike Fulcher is my colleague, and then Paul Owen Smith is his technician, and then all the individuals, both retired and current, that are working with us in regards to the research I just discussed. And thank you for your time. That is my email, matthewtancos@usda.gov, and my colleague, Mike Fulcher. Please reach out. And again, some of these situations, if you observe some severe dieback of some of these invasives I was discussing, please reach out, send us photographs, let us know, and we'll determine whether we can get out there and what all that entails. But the more eyes, the better. So thank you with that. And I'll be happy to take any questions.
Great. Thank you, Matt. There are a lot of questions. Okay. How do you choose which plants to target in your research?
Yeah, so that's a big issue. As I was saying, we have stakeholders from across the U.S., the rangelands, Western U.S., Eastern regions. A lot of it depends on who this, it's really relative. If there's something that has potential or a high economic, impactful invasive weed, that's going to be a big target right off the bat. These federally noxious weeds are big targets, something that is demonstrated to have high impact either to our ecosystems or agriculture. Again, we're the United States Department of Agriculture. So something that relates to agriculture, either directly or indirectly, either it's toxic to livestock or it competes directly with crops, that is what we're really geared to investigate. I know there's a lot of invasive weeds, but some of those that are mainly more maybe not as damaging, we might not have enough support to really validate all of the research and time to investigate agents for those. So it really depends.
But
yeah, if there's,
it doesn't stop people like Mark Imlay and Judy and I from banging on your door and saying, control these.
Yes. and that's the good thing with stakeholders. If there's enough support from the top that says, hey, why aren't you guys investigating these? So these organizations, you help lobby, you help support by contacting politicians, representatives, and having them say, well, are you focusing on this? What about this weed? And it kind of filters in that way to us. So it's all relative and it all depends. And then sometimes if we happen to find some agents that look good, just by luck, we're going to target that invasive weed, especially if it's a damaging, important weed.
My question was whether you know of any citizen science projects that those of us who are concerned about invasive plants, or, and particularly I love this biological control angle, that we could get involved with. Thanks.
Yeah, and I think Mark actually is one of the big proponents of, I don't know if it was with this organization, it was a couple years back, but I think he sent out an email about if you see something, say something. And that's the thing, if we have a lot of eyes, like that garlic mustard, this potentially new species, that was just luck that we happened about that. We saw that. It could be a nice agent. Maybe it's too broad of a host range. We don't know. But identifying those things is important. And obviously I can't be everywhere. We're limited to our staff. So the more people who see stuff is definitely beneficial to us. However, I'm not an outreach person, an extension agent. I can't identify all these things from hundreds of people. But if you identify something promising, like a severe dieback in any of those nasty invasive weeds, that would be well worthwhile to shoot me an email with photographs and a little background information to help me determine if it is really a pathogen, or maybe it's an abiotic event. Maybe it was a hard cold frost, or maybe someone sprayed pesticides and someone didn't know they did that. So there's a couple things you can do right off the bat that will narrow it down for me. But the more eyes, the better.
I was the one that said, if you see something, say something.
Yes.
And as I remind Matt, when you get a chance, please come out and see the massive die-off of mock strawberry, which is actually much more invasive than most people used to think. And it's actually all over the place, but in Swan Park, July of August, suddenly half of it dies off. So I invite you to put that on your schedule, have somebody, maybe you could send Jill out here.
Sure. Okay, thanks, Mark. We got a lot of questions. I'm going to keep moving, but if you have another one, pop back in. Okay, Patty McGrath, will you try to seed native plants to replace the dying invasive if the pathogen works?
So in that case, let me give you a better overview of that. So at the Agricultural Research Service, our job is to do the research to identify, evaluate, and go through that regulatory process. So once we get that approval to release, then we work heavily with our collaborators, state departments, county, park services, and they really take the lead to incorporate that into their integrated pest management approaches. So that might be part of their tool belt, is which they incorporate that. They also have other cultural practices and tools that they implement as well. So that's going to be really dependent upon that. But for example, with the Ramularia of the Croppina plant out west, so we're going to assist with that, and we're going to do more of the research aspect. How effective is it? What are we seeing? But we're not going to be really doing the extension aspect because that would be those local extension individuals who would be taking part in that. So it would definitely be part of that integrated pest management, but myself and my team are not going to be having that much of a hands-on approach once it reaches that stage.
Thank you, Matt.
In Montgomery County, the Weed Warrior program, when we
Yes.
do pull weeds, there has been situations where we have used seed or something to try to replace, but it's a little bit random. I'll just say that.
This is Jean Resgerda. Have you seen any plant-specific pathogens get released, then later mutate spontaneously to infect other native plants?
No.
We got multiple questions about this in the chat. We probably got like a dozen questions on this.
Yeah, so it's a hot topic.
Yes, and that's a valid question, especially, and that's one of the big reasons. So with like the EPA regulatory aspect, so that only applies to plant pathogens. So when groups release foreign insects as biological control agents, the EPA does not regulate that. So it's part of also a tendency of you using, it's a pathogen. It's a disease that's scary. It's going to have people concerned more than just an insect. But in that light, no, I do not know of any examples of a released pathogen jumping hosts and causing an impact like that in switching and being mutated. Now, is it possible? Anything's possible in nature. But you would have to have a lot of selective pressures to encourage that. And again, going through these rigorous regulatory review processes, that should be very minimal and it shouldn't be a real risk. But that's one of the questions I want to look at with pathogen evolution, is to kind of address that question head on. Because if you do have related strains that are specific, but there are other strains that can infect crops, what is the chance or likelihood that you can have evolution or recombination between the two? So that's a basic science question that you can ask and try to quantify what is the real risk of that occurring, as opposed to just a statement of saying, no, it won't happen. It would be a difficult question, but a very valid question and very applicable to our agricultural systems, better understanding how new diseases emerge. But no, I do not know of any evidence of an agent being released for the control of invasive weeds and then mutating to a new host. And obviously you can't test every plant in existence, especially in North America, but it's a process that you actually focus on. It's a centrifugal method that you focus on the most closely related plants to this invasive, and then you work outwards and you get more distantly related. And then you're also investigating those threatened and endangered plants that are closely related to that non-native, and also some agronomic crops that would be important in that aspect to evaluate. So you start close in that family, that genera, and then you work outwards and you get more distant related. And that is the best tool that we have to, without sampling every known species, to ensure that it is host specific. But based on biology and pathogen evolution, a lot of these times you're going to find it. It's going to infect another host if you evaluate enough of those closely related ones. Otherwise, it will be specific.
Okay, Angela Moxley asks, do pathogens usually receive regulatory approval? What are some reasons a pathogen might not be approved?
A lot of reasons why they won't be approved. One of the big ones is too broad of a host range. And we've had that several times with some of these foreign agents. My predecessors, they've submitted it, but it has a broader host range. And in some of these cases, you can make the argument, well, yeah, it could infect this plant, but this plant is all the way on the East Coast, and we're only going to release this in an arid environment in Utah. So there's no real risk that it could infect that plant over on the East Coast. Let's say it's in Maine on the coast. However, Fish and Wildlife, EPA, some of these regulatory agencies will still say, no, there's a risk and potentially it's a real risk. So stuff like that is very prevalent. So I would say a lot of the time you have to demonstrate very host specificity to these pathogens. Otherwise, they will be shot down and not approved.
EJ Scaruffi asks, why didn't these pathogens come in at the same time as the invasive plants themselves?
A lot of the time, it just depends. Some of these are like brought in, some of the seed are brought in on contaminated grains, or they're brought in as ornamentals. They're brought in accidentally. So again, we don't always see diseases everywhere out in the field, even in our agricultural systems. There is disease, but it's not always everywhere, because if it was so widely prevalent, it might actually kill the plant and suppress it. So in these native regions, a lot of these pathogens co-evolved. So they're not, it's in their best biological interest, not to kill your host. So in those cases, you might not have broad outbreaks. Maybe that summer, that year, disease was minimal, and that's when it was introduced. There could be a variety of explanations of why it didn't come with it, but sometimes it is introduced with the invasive. So Canada thistle was introduced to North America, and it did bring a rust along with it. So that rust is present in North America. It did not emerge in North America. It was brought with the host. So sometimes that does happen. Sometimes maybe diseases and pathogens are brought over, but they're not that effective. So they kind of die out, or they don't really thrive in the new environment.
Many of our wild areas and parks are overrun with Japanese stiltgrass. It would seem to me this is having a disastrous effect on those ecosystems. What is being done about this in the mid-Atlantic? There's not too much being done except trying to find something, and then mostly doing manual and chemical control.
Yeah, and I think that's one of the issues with the stiltgrass. And I know I have it in my yard on the forested margins encroaching in. Yeah, it's cultural practices, chemicals. That's all you really can do, to my knowledge. But I'm not an expert on stiltgrass and managing it from that aspect.
Someone asked about knotweed. If there's, could you address knotweed a little bit as far as pathogens? I put in the chat that there's the knotweed psyllid that is has been approved and is being tested and released. So there's an insect, but
yeah, we're currently evaluating. We have a pathogen that we're looking at knotweed right now, actually identified in West Virginia. And it showed nice disease in the field. So we're evaluating that in our greenhouse right now. We were able to recreate the disease, but we're still doing other trials on that. So
Good.
Too early to know, but there are pathogens.
Great. Okay. Have you investigated whether beneficial fungi can boost the vigor of native plants and plant communities enough to help them outcompete invasives and pests that wipe out plants and trees?
That is something, yeah, but that's not what our approach is. We're not to amend the soil or try to affect it that way. Our goal is to more target that invasive native and try to suppress those populations, because in that aspect, you will then benefit the natives by suppressing that population. So that's what our main goal is. But we are doing research looking at the microbiome, virome aspects to see if we can exploit some of those organisms, maybe they're not pathogens, that are co-evolved with the organism in the microbiome, and manipulating that into essentially making them less resistant to abiotic conditions or other biotic factors. So that is one avenue of why we're looking at the microbiome and virome, is to see if we could exploit that to work against the invasives.
The mycorrhizae are often specific to a particular species of plant. It's not like maybe they're 5 and all you have to do is give the native plants those 5. It would involve basically for every plant species determining what mycorrhizae would let that plant species really do well, and then use that mycorrhizal species with that specific plant species. So that's a really, really big task if you're going to take that route.
Ginger, Woolridge, are there examples of these pathogens being used successfully in the past?
Yes, especially so some of the colleagues over in New Zealand, Australia, they have a very more streamlined, I guess, approach to releasing agents and more of the biological control. They've released numerous ones to help manage invasive weeds over in their systems. But yes, in the US, I think we have a better profile of releasing insects as biological control agents because it's more of a streamlined process as opposed to pathogens that can take significantly longer. But we've had insects that were released that help manage this. But also like skeleton weed, I believe, was one of the first ones that a rust was released, and that worked very well. But in some of these cases, this rust was actually too specific. So it only knocked out a specific ecotype. And that has a downside because then you allow another ecotype or genotype to then take its place. So you want specific, but you don't want it too specific that you only infect some of those genotypes. And that's one of the good things we're seeing with this Croppina fungal pathogen is that it is known to infect all known genotypes in North America. So we're hoping that will be a better approach.
Is resistance an issue?
That can always be an issue. We see that commonly, both in the field and in agricultural systems. You can develop resistance. You have this warfare, essentially, if you want to describe it, between pathogens and plants. Developing plants have their own immunity. It's quite impressive of how they defend against a variety of microbes, let alone plant pathogens that are trying to infect them. So that is most definitely a possibility. I don't know if there's much evidence of seeing that, to my knowledge, of seeing an agent that was released and then resistance inhibited it or made it non-effective. I think a lot of the cases, especially with the insect side of stuff, is that they see insects become less effective, but it has more to do with the environment and climatic conditions. For example, if the plant is able to expand further northward out of the range of the introduced insect, it can't go with it. So it kind of escapes that pressure by moving in a way to new territory that the insect's not adapted to thrive in these colder conditions. So that has been demonstrated. I'm not sure of seeing resistance to a release pathogen for this stuff, this kind of stuff.
For the pathogens that are endemic that you're not introducing but are already here, why have they not already curbed the growth naturally on a larger scale of some of these invasives?
It's a great question. So a lot of that has to do with localization. In some of these cases, and for example, the fig buttercup, I've only seen disease in one small section of this infected forest. Sometimes you have these pathogens that emerge, and it's a recent emergence, a recent introduction, and they haven't had time to progress in a county or in a field, let alone statewide or across the nation. So that's where the idea of trying to target some of these endemics is that maybe they're not there yet, and you can exploit that by assisting with the dissemination. And not all pathogens are created equal. Some pathogens can rapidly disseminate hundreds of miles on their own. Others, they need rainwater and splash. So you can see how that would take a very long time to spread. So, and that's the idea of if we could harness that and utilize it to assist in the spreading, that it could be beneficial as even if it's an endemic.
Stacy McGraw asks, when evaluating whether a pathogen will only infect the target host plant, do you test it against non-native ornamental plants or only regionally native plants?
So if there's ornamental plants that are closely related that are of significant value and just ornamental plants in general, they would be evaluated. So again, it's that centrifugal method of anything closely related, you get a diversity of organisms. And this also goes through that whole regulatory process. They'll look at that list of what you evaluated with, and maybe Fish and Wildlife said, well, you forgot about this group, this threatened and endangered plant, or what about this crop? That could be, did you check it? And you have to go back and evaluate that. So there's multiple lines to catch that and to ensure that we're doing the physically best we can to demonstrate host specificity. And again, another caveat, especially with not like individuals who are not familiar with pathogens and that, is that so in species in general, some of these, like I mentioned, you can have a species that has been demonstrated to infect a variety of plants, a big broad host range. But within that, you can have very unique isolates or strains that are very host specific, even clades within that general species that are very host specific. So just because you see this name pop up and well, that's been demonstrated to infect this plant and this plant. Well, maybe this isolate, this strain is very host specific, and that's very common as well. So just think of that in the back of your mind. Just because you see a species that has been listed in other target plants, it doesn't mean it's that same isolate that will behave the same way.
I was wondering, what do you have a percentage measure of death, or can it not reproduce, or how do you decide that something's actually worth using, even if it's shown to be host specific and not gonna wreak havoc in some way?
So you're saying in the greenhouse aspect when you're evaluating, so if it's very specific but it causes okay disease, should we still progress forward? And that's really subjective. In some cases, if there's nothing else in the environment that's controlling this, it's a really nasty invasive, it might be worthwhile to try to progress with that anyways, because maybe it will do better in a natural field setting as compared to a very controlled, optimized disease greenhouse that you're trying to cause disease. So it kind of depends, and it also depends on the regulatory panels. Some of them might say, yeah, it's specific, but is it really going to do much damage? And is it worth it? Is it worth the risk of not knowing, even though it's not going to cause much impact? So it is very subjective. It's not like there's it reaches this mark, so yeah, it goes forward. It's more subjective if it can infect all those genotypes and if it shows potential to reduce biomass or seed set. I guess that would be the big one, is that you can significantly demonstrate that, how it can reduce biomass or seed set or growth in some way.
Fawn Palmer, do you want to unmute and ask your question?
I was interested in knowing whether you're looking at adaptive loci, and those would be, the genetic analysis of what is able to move and evolve. We've got climate change, which is changing a lot of the conditions, and I'm, we're not dealing with the static system. The native species are doing the same thing. Are you looking at adaptive traits that would allow the pathogen to more seriously or more widely infect?
So in the genomics aspect, yes, we're doing genome sequencing. We're looking at molecular aspects of the pathogen. And in regards to that, which I think you're referring to, yes, we're trying to better understand the evolution of the plant, the pathogen, and the relatedness to other pathogens that can infect agronomic crops or something similar. But ultimately, with that genomic aspect, is what we're trying to identify is the pathogen evolution and relatedness to other species. So that's what we're really digging at in that end. And through genomics and whole genome sequencing, you can kind of get an idea. You can see tails or landmarks or genes that have there's evidence that they were acquired through a different system. They're novel in that sense. So that was kind of the aspect of looking at gene flow and movement in some of these related pathogens to better understand that.
Linda Rieger?
I was wondering, I think somebody else asked this question. Once you find something, that works, who manufactures it, or who gets a patent?
Great question. There's a lot of work going into that right now, and a lot of that hinges with the EPA. So if you find an agent, whether it's endemic or a foreign one. If you want to apply it and have other people apply it as a biopesticide, which is a biological control agent, you would have to have it registered. So you would have to go through the EPA process to get it registered as a biopesticide to then be inoculated for that specific goal. So this is an issue right now with some endemic pathogens of Canada thistle, this rust. There were trials, and some of that work was done here at Fort Detrick, looking at this endemic rust, and you could apply it almost like a biopesticide at high concentrations year after year, and you would actually see plant death, and you could have eradication. However, the groups out in Colorado, they were doing that, but now they've got a lot of pushback from EPA because it's not a registered product. You do not have that approval to use it. Even though it's an endemic pathogen, you still are treating it as a biopesticide, and EPA controls all pesticides, also biopesticides. So you have to go through that regulatory process. That has happened with other endemic pathogens that were utilized more for commercial interests in the agronomic sector. You could have an agent, you get the EPA approval, and you have it registered, but a lot of those times there wasn't enough financial interest to keep re-registering that product when its limit went up. So it kind of just went to the wayside. So that is a big concern, especially in some of these agents like what we're working with. We're not going to sell it on the Lowe's or Home Depot shelf of products. It's going to be going to state departments, park services, landowners. So there's not a big financial patent that we're going to have because it's going to benefit the stakeholders that need it. So there's a lot of work right now going with the EPA and APHIS and trying to better clarify that and understand that, because that does hamper research efforts in that, because if you get approval and then I can't give it to the stakeholders,
Right.
how does that work? So that is a issue. And usually a lot of that would then go into the stakeholders who are going to do that. So a lot of the work we had with other rusts of invasives, such as in California, a lot of those individuals would, we would teach them the protocols, how to amplify and produce the rust spores. They could harvest it and then they could go release it on their sites. So you could have that. But again, that takes resources and funding. So who's going to sponsor that? Who's going to support that? A lot of it is dependent on the state and whether there's a potential financial interest from a company. But in some of these like big rangeland weeds, that financial interest might not be there to make it on a large scale. It's more there at the state level.
Catherine Pleasant, she was asking if there are any examples of successfully reaching the last step to have a release on a Maryland invasive.
Not that I know of. I think a lot like our predecessors in that were really focused on the Western rangelands because those were bigger stakeholders in the agronomic sector. But I think like right now, I know there is a group from West Virginia and Penn State, Verticillium wilt, a tree of heaven. And I think they're actually going through the process of EPA registration. So they're going through all of that process right now. But no, I might be wrong, but I can't think of anything off the top of my head for that. Good
segue from the tree of heaven. Ginger asked, do you think your group would ever deal with the vines that are killing our canopy trees?
It's a possibility. I know like some of these large trees, I'm trying to not get involved with because those are a nightmare to grow in greenhouses and they have a very long time frame of growth. And as a government scientist, I am required to have so many publications a year. So it makes it very difficult to investigate those very long-lived invasives such as trees and that. So yes, that is a possibility, but at the moment, those aren't ranking high on our stakeholder lists of being critical ones. What I've commonly seen, it's the annual ones that can really take off and spread very quickly and just rebound year after year. That's consistently popping up on these lists for the stakeholders.
So I think actually the Ginger's question was referring to specifically the vines that are killing trees. So things like bittersweet and, other types of vines like that.
Not at the moment, we don't.
Margaret Stanton says, scary to think we would bring in a foreign pathogen to deal with foreign plants. Really worry about the future damage to our environment. Not disrespecting the effort, but it's still scary.
Yes, that can definitely be opinions. Yeah, and that's really why I wanted to highlight the overview that goes into this process, because it is not willy-nilly. It is not like the cane toad release in Australia in the way back when you just say, here's something, let's throw it out there. It's significantly more to the process than just doing that. So personally, I am not fearful that something like that would occur, because I know of all the processes that go involved with that. So that's what I was trying to demonstrate to you all, so you're a little more calm and everyone understands what actually goes into this.
Mm-hmm. Yeah, and there's no 100% guarantee, because it is the laboratory of the planet. It's,
Yes.
once it's out there, that you find out. But so far, as long as all the precautions are taken, and there are really a lot of hoops to go through, it seems like we either do that or we just spray pesticides all over
Yes.
the place. It's kind of like, trying to find something that works better.
I'm really excited, and I suspect Matt will be excited to know that there's a good discussion about which legislators we should be talking to. So about, first of all, a sympathy for the fact that there's only 4 of you working on this. Like, we recognize that this is understaffed, underfunded, and we need to throw a lot more at it. And so I think there's some conversation about who can we talk to at the legislative level, that kind of thing. So that's hopeful. So I say, yes, go write letters, talk to your Congress members and your state people and everybody else that you can talk to, and get this ramped up, because it's a huge issue and we need more mats.
Matt, maybe you want to do a final, just a summary of, again, if people find what looks like it might be a pathogen, how should they communicate with you? They obviously have your email, but maybe you could summarize that before we sign off.
If you identify any of those weeds I was discussing today, if you see some dieback when you're hiking, when you're on your own land, anything like that, if you make note of that, take pictures and give me as much information as possible, so then I can go through that and try to determine whether it's worthy of going out there to the trip, to evaluating it. Maybe it's actually just a abiotic thing. But yes, if you see disease, if it's very evident that it's a pathogen or a disease, please let me know, especially on those high profile invasives, such as the wavy leaf, fig buttercup. Poison hemlock is another one that I'm always trying to look at. And so a variety of those, please let me know. The more eyes, the better.
And I'll just add really quickly, those of you who know about the early detection distribution mapping system and the phone app that is still going right now, called Maiden Mid-Atlantic Early Detection Network. When you're out, if you see something, you can report it using that. You report the plant, and you can make a note, take pictures of the symptoms, and say, you're wondering what this might be on this plant. And that way it gets tossed into the early detection distribution system that is nationwide. And people like us, well, some of us are verifiers. We get notified when reports are made. And as we get time, we check in on them. But just an added way to report it.
One thing that I just wanted to add is, well, I understand and very much appreciate everyone's concern about the potential for impacts on non-target species by releasing biocontrol agents. I also want to make sure that we think about the fact that there is like a never-ending supply of new invasive plants coming in. I mean, whether it's through just accidental introductions, the nursery trade, whatever, it seems as though we're being a lot less careful in terms of what plant invasives are coming in, rather than the concern about the biological control agents we release. So we just have to, you know, consider all of the competing risks that we're facing, and intentionally releasing a biological control agent shouldn't be done lightly, of course, but with all of this regulatory, you know, process that is required, I'm not nearly as worried about that as I am about new plants that are coming in.
Great point. Mhm. Okay, Merity Ware, and that's the last one. Go for it. You have to.
My question was answered. Judy said the name of the app you just mentioned was called Maiden, Maedn. So that's
Okay.
great. Thank you. Great
Okay.
presentation.
Thank you, Matt, for a fantastic talk and lots of great questions from our wonderful attendees. So, yay, thank you so much. Really appreciate it.
Yes, thank you all. Great time. Thank you. It's always good to see people interested.
Yeah.
Thank you.
And we'll be letting you know if we find things. So.